Hello, my name is Marwa Elsayed, and today I will be giving a brief introduction over our study. So, in our study, we wanted to further explore cilia and flagella, and we wanted to look into how differences in their lengths may impact human health and associated disorders. So, we used the organism Chlamydomonas in particular because they have flagella that is structurally identical to cilia, and they're also able to undergo mutations that other organisms would perhaps be lethal for them to undergo. So let me get my pointer started over here. So we first took wild type cells and our mutant cells, which are called SHF1 cells, and we grew them, we fixed them, and we looked underneath a microscope to look at their cell body volumes and their ciliary length. And so up here in figure 1, we see orange represents our mutant cells, blue represents our wild type. As we are moving down this horizontal axis, we see cell body volume increasing and then frequency up here increasing. And so we see that our mutant cells have a greater cell body volume than the blue, which is our wild type. And we confirm this over here, showing that the wild types are about double the size in cell body volume than our wild type, and that is statistically significant. So this tells us that the mutants have lost their ability to regulate their body cell body size. And so we then further assessed, as mentioned before, the lengths. And so we can also see how the mutants have a shorter length than the wild type. And so to take this further, additionally in the literature, there's a report on how acetone can affect cilia and push them into utilizing the Krebs cycle in order to survive. And so down here, we can see we took the short mutant cells and we incubated them in 30 millimolars of acetate for varying times. And so as you can see, as a result, with increasing time in the acetate, we see a decrease in the number of cells with flagella. So they are in fact unstable when we add the exogenous acetate. And so we were also interested in seeing what other differences there were between the proteins. So we used a global proteomic approach, and we grew the cells to equal density of cell number, and at the same time we isolated flagella over here and then cell bodies and subjected them to protein sequencing. So over here in figure 4, we see the total cell bodies and the total flagella. We identified the number of cell body proteins that were specific to the mutant, which was 426, and then specific to wild type, which was 1,293, and then in the middle is the commonalities that the two shared. The proteins had in common between one another. And again, we did the same thing for the flagella over here. And so we then identified how many out of the total proteins found for cell bodies and then the total proteins found for flagella were statistically significant. And so we can see that down here in figure 5. So going back up to figure 3, it shows this proteomic approach that we use to be able to harvest the cells and separate the cilia from the cell bodies. So since clammy are able to shed their cilia in acidic pH environments, we placed them in these conditions and centrifuged them and separated the cell bodies, which were heavier than the flagella, and were able to get the purified forms of each, as we saw in figure 4. So moving forward with this project, we plan to take these statistically significant proteins and further examine them to differentiate between these two strains and learn more about the flagellar assembly and their functions.